Okay, I think we're all set with that. Last month, we had a presentation from the doubly honored Dr. Robert Michael Pyle, who is an honorary fellow of both the Royal and American Entomological Societies. Although there are only 8 such scientists who have been awarded both of these honors, we are very fortunate to have another such doubly honored fellow this month. It is my great pleasure to introduce Dr. Lynn Riddiford. She has so many honors that I will only mention a few more. I think her biography has been distributed. She's a National Academy of Sciences member and also a fellow of the American Academy of Arts and Sciences, as well as a member of the Washington State Academy of Sciences. Dr. Riddiford is a professor of biology emerita at the University of Washington, and she now conducts her research at Friday Harbor. Lucky you. So the title of Lynn's presentation is Unraveling the Mysteries of Metamorphosis. And since more than 85% of insect species undergo complete metamorphosis, this is a topic of great interest to all of us. So thank you so much for being with us tonight, Dr. Riddiford.
Well, thank you. And glad to sort of see most of you as the pictures come up. But anyway, what I want to do tonight is I want to tell you sort of history of some of the hormones that control metamorphosis. And we're going to talk only about 3 hormones: the brain hormone, or PTTH, the ecdysone, which causes molting, and juvenile hormone, and go into how people discovered them. And then I'll bring you up to date to what we know today, little bits and pieces. And I'll have places in the talk where I will ask for questions, and then Paulette will take them out of if there are any in the chat. So let's go. Whoops. Now, well, let's see. Okay. So insects, of course, as you probably all know, we have different kinds of metamorphosis. We have the ametabola, the silverfish, which really don't undergo a metamorphosis. They just gradually and eventually, after many molts, will go into the adult stage, and then they keep molting as adults, just like the crustacea from which the insects arose. And then we have the human metabola, and this I take the milkweed bug, and of course, then the complete metamorphosis, and here my favorite, the tobacco hornworm. And why do insects molt? They have to molt in order to grow because their cuticle is their exoskeleton, and they grow within the instar as they feed, the nymphal or larval instar, but they can't grow. The cuticle can unfold a certain amount, and then they have to molt. And they have a certain number of stages, which we call instars, like here in the Acapeltis, the first, the second, the third, the fourth, and the 5th instar nymph, and then that goes directly to the adult. But what I'm going to talk about most about today are the complete metamorphosis. That's in like the tobacco hornworm, where you I have to see where the egg is. I can't see an egg somewhere on there. But you have the 1st, the 2nd, the 3rd, the 4th, and the 5th instar. Then it climbs down, digs a burrow in the soil, pupates, and then comes out as the adult. And as I say, this is all controlled by hormones. And these are the 3 hormones that we're going to talk about. And I'm going to talk about prothoracic tropic hormone, which drives the prothoracic glands, is PTTH. I'm not going to say that. And And that gives you ecdysone, and ecdysone causes molting, and ecdysone could cause metamorphosis. And why you don't get metamorphosis somewhere here is because the nymphal or the larvae have juvenile hormone, and that will inhibit metamorphosis. So we want to know what's happening here. And let's look first at the brain and the prothoracic glands. The brain, and if you look at a silk moth brain, this happens to be a Cecropia brain, I believe, or else a Polyphemus brain. You can look at this brain and you see some cells here. These cells turn out to be neurosecretory cells. They're big cells. They have a lot of protein in them, this neurosecretion. And that protein, in one case, has actually been identified from the silk moth, bombyx, to be what they call bombyxin, which is an insulin. I'll show you that in a second. And PTTH, you can't see those cells here. There are only 2 of them on either side of the brain, but here's a diagram, and they send their axons down into, down into the corpora allata here, and then they release their hormone into the blood, and PTTH then comes into the prothoracic glands, which are in the thorax, and I'll show you a picture of those in a second, and that causes them to release ecdysone at the time for the molt. If they're in the larva stage, juvenile hormone is being released by the corpora allata at certain times, and we'll see the titers of the hormone in a minute. Now, a long time ago, in 1918, actually, Kopech in Poland was looking and trying to understand metamorphosis of the gypsy moth. And he was good enough to go in and take the brain out of the head of the gypsy moth larva. And he used female larvae in the final instar. And they took about 10 days or 11 days in the final instar, feeding and growing. If he removed the larval brain on day 7, he wouldn't get a pupa. They lived, they didn't die. They just lived and grew bigger and just never metamorphosed. But if he, if he also did ligation experiments where he tied off the head, and of course they couldn't grow at all since they couldn't eat. But anyway, if he did the same experiment, either removing the brain or ligating on day 10, then they went ahead and made a pupa. Of course, it would be a brainless pupa. But even in those days, he postulated in his 1922 paper that the brain was giving off a substance into the blood, which at that time, they basically knew that tadpole, that thyroid hormone, if they used extracts of thyroid, would cause tadpoles to metamorphose early into frogs. And so they knew about hormones, and he said, okay, this is a substance, a hormone that goes into the blood and it acts on some other tissue. He didn't know what it was, and that will cause metamorphosis. 50 years, 1980s, about 50 years later, no, 70 years later, sorry. 2 Japanese isolated these 2 hormones, the bombyxin, which is an insulin-like molecule. It looks like very much like insulin in us. And then PTTH, which is a much bigger protein and nothing, not insulin-like at all. And if you look at, and this is staining, taking for the messenger RNA, looking at the messenger RNA for these tissues. And for Bombyxin, you see it's in those center cells that I pointed out before. And for PTTH, it turns out, only 2 cells in either side of the brain. Now, in the 50s, Carol Williams at Harvard worked on Cecropia. We're going to talk a lot about Cecropia. And it was his favorite insect. And he did ligation experiments and basically repeating what Kopek had done, except that he looked at both the head, the thorax, and the abdomen. So he tied strings around the neck and between the thorax, well, actually between the first and second abdominal segment here. And if he did it early in the last instar, they had no PTTH and they didn't metamorphose. If he did it midway through the feeding period, or slightly toward the end of the feeding period, he got the head would fall off, die, but the thorax would pupate. And so that says that enough brain hormone, PTTH, had been released to turn on the prothoracic glands, but the prothoracic glands hadn't released enough ecdysone at the time of the ligation. So the abdomen stayed larval. And then if you did it late, after when the animal stopped feeding or just before, then what you found was that you got pupation of both the thorax and the abdomen. But the ultimate experiment that he did was to take an isolated pupal abdomen, take out the gut, so it didn't, it was empty, but it still wasn't. So, and then he implanted brains, active brains that he knew would have the PTTH and prothoracic glands into this isolated abdomen. And he got a nice moth abdomen here. So this kind of simple experiments were really outlining this and the neuroendocrine system here. So we can sort of summarize this part, that the brain gives you the PTTH, those neurosecretory cells on either side of the brain, that activates the prothoracic glands, gives you molting hormone, and you get the molt. And now I can stop if you have any questions.
Yeah, so one question is, did Williams know about the PTTH and the ecdysone, or was he just going on functionality rather than understanding what the chemicals were at that time?
He didn't know what the chemical was. He knew that the prothoracic glands, because of previous experiments in the 40s of some Japanese that had done ligations on the silkworm. And he knew that the prothoracic glands were responsible for what the hormone that was causing molting. He didn't know what the hormone was. And they called it ecdysone at that time, but they didn't know what the chemical nature of the hormone.
And they knew the brain was also important.
And they knew the brain. I mean, he knew from Kopek's work that the brain was important. And we'll get to Wigglesworth in a minute. And he had done his work in the 30s and 40s about juvenile hormone. And so everybody knew that that these hormones were involved, but nobody knew what the chemicals were.
Okay, another question is when were Carol's earliest Cecropia brain experiments performed?
In the late 1940s. And that experiment that I showed you in the previous slide was actually published in Scientific American in 1950. He never published, some of the papers that he wrote were later 52, 53, but I think he knew a lot about the brain, and he was doing experiments on the brain because in Cecropia, they diapause during the winter, the obligatory diapause, and it was, he had found that chilling the brain, all you had to do was chill the brain. You didn't have to chill the whole animal. Chill the brain and put a chilled brain into a non-chilled animal, and it would develop into a moth. So.
Okay, why don't you press on?
Okay. So now we go to Wigglesworth. Now Wigglesworth was in England. He was at the London School of Hygiene and Tropical Medicine, and his favorite insect was Rhodnius, this blood-sucking bug from South America. That's the vector of Chagas disease at trypanosome, and it has to have blood. It has to feed on blood in order to molt. As a nymph, every instar nymph, it has to feed once in order to get the molting to go. So this is why it's a good vector for disease, because the second feeding, it'll excrete the trypanosome and into the host that it's feeding on. Anyway, and in this case, this is the epidermis. Insect epidermis is a one-cell layer thick, and it makes the cuticle that lies above it. And so when they feed on the blood meal, then they'll start the molting cycle. And so after, in the case of Rhodnius, when he did ligatures between head and thorax at different times, he found that that he couldn't detect anything that was happening before day 6 or 7, because that would all prevent molting. But after day 6 to 7, then the animal could molt without its head. And but you wouldn't see the new cuticle until about day 13 here, and then they would ecdyse day 13, 14, and here's the old cuticles taken off, and you get the new cuticle. And the cells, you notice, are in a different conformation that they go through in this molting cycle. So he decided he'd do some decapitation experiments, and he did it on nymphs so that they fed from the blood in 6 days that they would molt. It depends on what stage you're in as to whether it takes 6 or 13 days. And in this case, these are small animals, so that decapitate if within 3 days of feeding, they just wouldn't molt. But if did it afterwards, then they molted. Well, that was very good. And then he developed this technique of parabiosis, where he put 2 nymphs together with a tube in between, cutting off bits of their head or all their head, depending on what experiment he wanted to do, and connecting basically the blood supply, the hemolymph from each animal would circulate. Actually, I think when they do these things and they put the tube in there and wax it in with just paraffin, then they squeeze the animal to keep the blood going between the two to make sure that it happens. And anyway, if you took a unfed, decapitated animal and you hooked it up to a fed one after 3 days so that it had the ecdysone, and then you would find that this unfed one would molt along with the fed intact one. So again, here he was showing that there was some circulating factor from the brain that caused, and at the time, that he did these first experiments, he didn't know again about the prothoracic glands or what they were doing, but within about 5 years, he did that. But it turned out that there was another organ in there that he had to worry about, and that was the corpora lata, which produces the juvenile hormone. And just to skip ahead, I'll just tell you that the hormone is present in the first, the second, the third, and the fourth instar nymphs, but the gland stops secreting the hormone in the last nymphoinstar. And here is Rodnius, fifth instar nymph, the normal adult, and a supernumerary sixth instar nymph, which are important for all of the experiments he did. Now, this is complicated, but I'll try to go through it slowly. So one of the experiments he did was take a fourth instar nymph and a fifth instar nymph and put them together. In this case, he fed it and then decapitated after 1 day. And that means that it wouldn't have released the PTTH and it wouldn't have released the JH because both the brain and the corpora lata are here in the head. So when he decapitated, he would decapitate here. But the other one, the fifth instar, remember that doesn't have any juvenile hormone, and they let that go for 8 days before he decapitated, and then that would molt to the adult, so that the animal here, the fourth instar nymph, also molted to an adult because there was no JH there. And the other, he did many of these different kinds, but the other that's really prescient for this is that if you took that fourth instar nymph that had the JH and fed it, and then waited 6 days, and then just removed the tip of the head. So it had no brain, but it had JH, and that was parabiosed to a fifth instar nymph who had been decapitated after 1 day, so it had no PTTH, no JH. And when they parabiosed these, then this fourth instar molded to this normal fifth instar, and the fifth instar molded to a supernumerary sixth instar. And so he then said that this was juvenile hormone and named it juvenile hormone by 1940. Well, Carol Williams came along in the late 40s, again, working with Cecropia, and he had always liked Cecropia, and of course they don't feed as adults. So he thought, well, he could use this technique of parabiosis and parabiose a moth to a pupa and make the moth live longer because the pupa is just full of fat body and nutrients. And that is true. However, it turned out that if you parabiosed the Cecropia moth to a Cecropia or to a Polyphemus pupa, or even just the moth abdomen to the pupa, that the moth would, the abdomen or the moth would live longer, but the pupa would molt to a second pupa. And in fact, the parabiosed abdomen would molt, make another cuticle, but it couldn't make scales because the scale cells have died. So it would just molt again in this kind of situation where the ecdysone is basically coming from the pupa. So, and so he said, well, okay, then juvenile hormone is the status quo hormone. If you give it to a pupa, you get a second pupa instead of the adult. And he did a lot of these types of experiments of parabiosis and of moths to pupae. And he found that the only source of juvenile hormone was the Cecropia moth male abdomen, not the female, and the Cynthia's moth abdomen, again, male, not female. Polyphemus didn't have it, nothing else had it, which, I mean, that was just a serendipitous result that gave us the first juvenile hormone extract, because all he did was he would dump these Cecropia male abdomens into ether and methanol and grind them up and get what he called a golden oil. And then he made that extract, and he took that in 1955 to 56. He went on sabbatical to Wigglesworth Lab at Cambridge, and he took that extract and he characterized it. He didn't chemically characterize it. You know, we found out that it was lipid and what it wasn't and what it was, but he also tested it out on lots of different kinds of insects. And the one I like is, I think probably Wigglesworth did the painting, but either Williams or Wigglesworth painted the golden oil of Cecropia abdomen extract onto a Rodnius, a 5th instar nymph Rodnius, and let it molt to the adult. And here you see the adult cuticle, except in the initials VBW that are in the typical nymphal cuticle on the abdomen. So now we have then, here's the Cecropia pupa with the brain, the corpora allata here, and the prothoracic glands that sort of are like bunches of grapes around the trachea in the prothoracic spiracle here. And now, so you have questions before we go on to more recent things? Okay.
Sorry, excuse me, I'm sorry. So at that point, Williams was speculating that his golden oil contained some substance that he was calling juvenile hormone that basically suppressed metamorphosis, even though he didn't know precisely what it was. Is that right?
That's right. That's right. And he did, and nobody knew. I mean, he published a paper in '56 calling it the juvenile hormone of insects and explained what he had done on these, but he didn't. And that was '56, and it wasn't until '66 that the chemical nature of it was discovered. And it was new to science at the time. I'll show you that in a second. In fact.
So he looked at, he used the golden oil that contained the juvenile hormone and found that substance worked to suppress development of a number of different insect species. Is that correct?
That's right.
Okay.
That's right.
So Bob Pyle is weighing in and saying Wigglesworth, surely the best entomologist name ever.
Yes. Yeah, that's true.
It made me laugh. Okay, well, let's go on now that now that we're on to juvenile hormone.
Okay, well, I just want to have a little insert here in about the growth regulators, insect growth regulators, which are most of them are juvenile hormone derivatives, not derivatives, analogs and mimics. And it all started, well, when Williams published the paper in 1956, he suggested that juvenile hormone could be used as a insecticide because his reasoning was they couldn't get resistance to their own hormones. Now that is not quite true, but anyway, but that's what he postulated. And then in about 1964 or 63, Karol Slimak from Czechoslovakia came to Williams' lab at Harvard and brought his Pyrrhocoris, these insects from that feed on linden seeds. And he put them in, to rear them, he liked to have them in big gallon jars and put in some sort of paper for them to walk up, because they, in the wild, they're found on the sides of trees. The seeds are at the bottom, but the Pyrrhocoris bugs are usually found on the sides of trees. So anyway, he put in paper towels because it was cheap. And these things did not metamorphose. The 5th instar didn't go to the adult. Instead, it went to a 6th instar, and sometimes it went to a 7th instar nymph, just supernumerary, and they didn't metamorphose. And so he thought it was juvenile hormone flying around in Williams' lab. So he even got some sent to his house, apartment in Cambridge, and raised them all there, completely not, and still they didn't metamorphose. And for about 6 months, it was, well, what is it? They finally tracked it down to the paper towels. And so Williams and John Law in biochemistry then extracted the paper towels to see if they could get whatever they would get out of the paper towels. And they made this column of paper towels, and then they poured different solvents through it. And what they found was, yes, they could get an extract of paper towels. And so Williams calls up the Scott paper towel people, and he says, what do you put in your paper towels to make it? And of course, they didn't want to tell him anything, but he finally told them what he had found. And they said, no, they don't put anything because they want to make paper towels cheap as possible. And so they just use wood pulp and whatever processing chemicals. But anyway, so it turned out that this substance that mimicked juvenile hormone was a particular compound in the balsam fir. It is not found in paper in Europe, because this paper in Europe is made from pine. It's not found in most other trees. It's specific to the balsam fir. And it's a juvenile hormone-like compound, and it works only on Pyrrhocoris and that family, Pyrrhocoridae. And it doesn't even work on milkweed bugs, the Lygiidae. Next, so that said, oh, here we have possibly a specific, very specific kind of compound that will work on a particular group of insects and not on others. Can we find other ones? And that started a whole new industry of these insect growth regulators for selective control of insects. And a whole company, Zoacon, was founded to do this, and they had different compounds. They had one that they could feed to the cow, and it would come out in the cow pat. The stable flies would lay their eggs, and they would not be able to come out and come out from, they wouldn't come out as flies. It also happens to affect embryos. If you treat Pyrrhocoris, adult females or the eggs, you get very strange unhatched embryos. It works on mosquitoes, on fleas, and so you have, even today, juvenile hormone mimics as insect growth regulators in most flea pollinators and flea collars. That's the active ingredient. It's also works on mosquitoes, and anything that has a adult pestiferous stage is the adult, it's very good. It's not good for caterpillars because, of course, it would just make them grow bigger and you eat more. So what people want to do is find an anti-juvenile hormone or something that prevents its action. And that has been very hard to do. So now the chemistry, and I don't think you need to worry too much about the chemistry, except to know that ecdysone is a steroid. It's like estrogen and testosterone and cortisone and all these steroids that you've heard a lot about. And it's a molecule, it's a lipid kind of molecule that can go into the cell. And juvenile hormone is a sesquiterpenoid. And as I said, when it was isolated and it was new to science as a chemical. And it's only found in insects and crustacea as a natural hormone and not in vertebrates. And doesn't seem to work on vertebrates.
So Lynn, before you go on, there are a couple of questions, specific questions about the chemicals. Do you mind taking a few questions about those?
No.
Okay. So we're looking at the structure of the juvenile hormone there, this bis epoxide. It sounds kind of unstable. It sounds kind of an interesting compound. So one question is how across how many species is that particular structure of utility, and are there other types of juvenile hormones with somewhat different?
Yeah, JH3 here is the main hormone in everything from the silverfish to even to Drosophila. But JH3 bis epoxide is only found in the higher Diptera. Not even, I think, in mosquitoes. JH3 is the main hormone of most insects. However, Cecropia and the Lepidoptera have JH1, which has an ethyl group here and an ethyl group here, and JH2, which has an ethyl group here and a methyl group here. So and that's only in Lepidoptera. They have JH1 and 2, and they also have JH0 in the embryo. And so that's specific to the Lepidoptera, and I don't think anybody knows to this day why that is true. JH1 seems to work on most other insects as well. It seems to be actually in some insects, like flies, is more active than JH3, which is one of their normal hormones. In bugs, in Hemiptera, it's a skip bis epoxide. So I think it's epoxide here and epoxide here, rather than a bis epoxide. Rather than this bis epoxide, it's a skip bis epoxide. And in Rodnias, that's what it is.
Okay, that's helpful. There's another excellent question about the JH feed, the feed with the juvenile hormone in it. And the question is whether dung beetles are also susceptible.
I'm sure they are. And I know that they worked on several beetles because they've used it on tenebrio so that they got bigger mealworms for fishing. And so, but no, I don't know for sure. I have to look it up whether anybody has really done work on dung beetles per se, but it certainly would. And I don't think, I mean, they did those experiments, but I don't think they use it routinely anymore. And maybe that was a reason that it messed up the dung beetles.
Yeah, because it seems like it would be a big change of the entire ecology if the
yeah.
if the dung beetles were taken out. Okay, well, why don't we press on then? Sorry to interrupt.
Okay, well, that's all right. Now I'm going to get a little molecular and talk about how hormones act in the cell. And basically, these two hormones, ecdysone and juvenile hormone, being lipids, can go into the cell through the cell membrane, and they bind to the receptor in the nucleus, and they will activate or inactivate genes. And how that works is you have a gene, and it's going to make messenger RNA that makes protein. And what turns on or turns off a gene there are some general transcription factors ahead of the gene itself, and there are special transcription factors. And special transcription factors can be ones that different genes have different transcription factors. So the general transcription factors are there probably for most genes, but the special ones can turn on and off genes. And some genes have a hormone receptor, what's called a hormone response element, where the receptor for the hormone binds to the DNA, and then the hormone is on to it, but it'll only bind to the DNA when the hormone's on. In other cases, it can bind to the bind to the response element before the hormone's on and inhibit the gene, and then the hormone comes on and activates. And ecdysone works that way. So the ecdysone receptor can be on the gene without ecdysone on it and inhibit it, and then ecdysone comes along and activates it. Now, so ecdysone was the first steroid hormone to actually be shown to act on the DNA. And the reason they could do that was because the midge and the higher flies, Drosophila, Proliferation, they have polytene chromosomes. And so as the larval salivary glands grow, the chromosomes just keep adding DNA strands in parallel, and many replicated ones. And then when a hormone or something activates a particular gene, what you see is a puffing out of the chromosome right here. And what that puff turns out to contain is messenger RNA. It's the actual visualization of the messenger RNA, because you have so many strands and it's going on in all of these strands. But anyway, so Klaver and Carlson was the one who first isolated and identified the chemical ecdysone. And he Klaver and Carlson showed that if you put the salivary glands of midges or flies into ecdysone, within 15 minutes, you got the so-called early puffs at certain sites. And these turned out to be special transcription factors. And then they went ahead, and these transcription factors would then activate other sites on the DNA to give you tissue-specific proteins, the late puffs. And that they showed that this would happen even if you inhibited protein synthesis, but you would not get the second if you inhibited protein synthesis. Well, Michael Ashburner then took Drosophila in 1967 and started looking at the puffs Drosophila. And by 1974, he had a scheme where ecdysone with its receptor, at the time they didn't know what the receptor was, they just called it the ecdysone receptor. We now know that it's ecdysone receptor, and it's a dimer of two molecules that are very similar. And you would get the early puffs in 10, 15 minutes. and you would have certain of these early puffs, different transcription factors. And then these transcription factors would activate the late puffs. They're tissue specific. And these transcription factors would also inhibit the early puffs. So this was sort of the so-called ecdysone cascade of action. And so if you look in Drosophila, here is ecdysone for molting, for the peak of ecdysone for molting from the second to the third instar, another peak of ecdysone at the time of puparium formation, and another peak when it goes from prepupa to pupa. And what you see here is a whole bunch of different transcription factors, all DNA binding proteins, and they're activated at different times in response to these big peaks of ecdysone going down here, including the ecdysone receptor itself. And you just had this cascade, and it just told you that you had a lot of different things being activated in this time. Now, if we go to Manduka, what we were interested in this transcription factor broad, because what we found in a fourth instar larva was, which had ecdysone acting in the presence of JH, that there was no broad being expressed whatsoever. But when you had ecdysone acting in the absence of JH here, the time of pupal commitment, which caused wandering behavior, it also turned on broad expression. And this is what's being shown here. There are different broad isoforms, broad protein, that this is the RNA for these proteins. And what you see here is that broad turns on in response to this ecdysone, and then keeps on through this big peak of ecdysone that causes the formation of pupa, which happens in the presence of juvenile hormone, so that you go to a pupa rather than to an adult. But if you give JH at this time when there isn't JH, let this peak of ecdysone come up in the presence of JH, you don't see broad appearing, and you end up with a giant 6 instar larva. If, however, you look at here, when the pupa goes to the adult, there's ecdysone acting in the absence of juvenile hormone to get the pupa to the adult. If, however, you give JH at this time, so ecdysone comes up in the presence of juvenile hormone, what you get is a second pupa, and what you see is broad RNA comes back up during this time when it normally is absent. Well, at this point, we couldn't do much, because if we didn't have a genome for Manduka, it's very hard to do genetics on Manduka, and we couldn't look at what was going on, trying to figure out what was going on. So we went to Drosophila. Now, Drosophila develops slightly differently than Lepidoptera in the sense that all the adult comes from a vaginal disc, at least all the head and the thorax, the eye, the head, the back, the wings, the legs, all come from a vaginal disc that in the larva just proliferate. The abdomen of the adult is made from histoblast nests, which in the larva of the fly are completely quiescent and only start to proliferate in the pupa at time of metamorphosis. And so what you see then in the fly is, if you look at the ecdysone titers, there is ecdysone for every molt and to make the pupa into the adult. And what you see is there's a JH, the exact peaks and valleys has never been discovered actually. But we know that it goes down here in the last instar larva and then comes up at the time of proliferation. When it goes down and ecdysone comes up here, broad is turns on, and then it's on all during the prepupal period and into the pupa. And this shows you what normally happens, that when you're making a pupa in the first 12 hours here after proliferation, this is pupal cuticle and head, thorax and abdomen, and then broad is absent during adult development when adult cuticle is being made, and you get a normal looking fly. If, however, you give JH just after this peak of JH at this time, or any time during this little period right in here, you get a different kind of animal. You get a mosaic. The head and thorax looks more or less normal, but the abdomen turns out to be pupal. And what you see is that there isn't much broad in the head and the thorax, but in the abdomen, broad has turned back on when ecdysone comes along. And so that you make a pupal abdomen and you don't have much adult cuticle at all. And so how does this happen? Well, there are transcription factors involved with JH action as well. And Tom Wilson first found a mutant in Drosophila, which he mutagenized his flies and then gave them huge amounts of methoprene, a JH mimic here, in their food, and picked out those that were resistant. And he got this mutant, and he thought it was a mutant that would, it was a JH receptor, but it took a long time to prove that. And it wasn't until Konopova and Jindra here suppressed MET using RNAi. This is a way that you can suppress a particular gene in beetles. It doesn't work very well in Lepidoptera, unfortunately, but in most insects, it works pretty well if you feed it to the animal, or if you give it to the adult female, you can get it through the egg. And you can suppress particular genes by this RNAi. And what they found in beetle, the tribolium, is that if you suppress MET, then you get precocious metamorphosis. This is controlled, this is the suppressed little animal. And then Cheika Minakuchi, and this is a picture that Kiyoshi took. It did the same kind of experiment with pupal homolog suppression by RNAi, and she got also precocious metamorphosis. So it said that both MET was somehow involved and pupal homolog may be involved. Well, what happened? And Marek Jindra and his lab went and really looked into the biochemistry of MET, the MET gene, and it turns out that one part of the gene, one part of the protein binds juvenile hormone, and another part binds to DNA. It's a transcription factor that can bind to this particular hormone response element. And one of the genes that the juvenile hormone activates is pupal homolog. So what they came up with is that the MET is a dimer molecule. When JH comes along, the dimer breaks up, JH binds to the MET, and then another cofactor protein binds, and the two met and tie bind to this JH response element and activate pupil homolog. And so, are there questions?
Yes, we have questions. So this is a very complicated story. So I just want to make sure that I understand it.
Okay.
And there are a couple of other questions too. So when ecdysone is released from the prothoracic glands with stimulation from the brain, then transcription factors are generated that control the gene expression. Is that basically what's happening?
Well, ecdysone goes into the cell, and the receptor is ecdysone receptor is in the cell.
Okay.
It can go into every cell, but if the cell doesn't have the receptor, it'll just go back out again.
Okay. And so a specific question is looking for the gene to activation sequences that puffs that create the activator proteins?
The puffs, yes, they could. They a lot of the puffs, the early puffs all turn out to be transcription factors. Well, no, I'm sorry, there's one that isn't, but all the others, as far as I know, all the others that come up within 15 minutes of exposure to ecdysone are transcription factors, which then go and activate different genes that can activate different genes.
Okay. And another question, when do the imaginal discs proliferate in Manduca?
The wing disc and the genitalia disc and the little imaginal cells in the larval legs are proliferating during larval life, and the wing discs are the most easy to look at, and they just proliferate. And then in the 5th instar in Manduca, once they start feeding, if they don't feed, they don't do this, but if they feed, then they start what we call morphogenetic proliferation. Instead of just proliferating as discs, they start to not evaginate, but make a disc that will be able to evaginate in the prepupal period. And the eye does the same thing at that time. And so it's in the first 12 to 24 hours after in the 5th instar that it's all happened, it starts happening. But it isn't until they start to make a pupa that then they evaginate and make the pupil wings and the pupil legs and the pupil eye. And that's why you have to have juvenile hormone during that prepupal peak of ecdysone in order to prevent those imaginal structures from overshooting. And in Cecropia, they really do overshoot. So you get an adult eye, with a pupil abdomen. Williams did this, taking out the coprolata during the prepupal period.
So despite all the complexity of what you're describing about the metamorphosis, it seems like there are some elements that are the same. We've been hearing a lot about messenger RNA as part of the COVID vaccines. I'm assuming that's the same.
It's the same thing, yeah.
Okay. Yeah. Okay, great. Well, let's go on then. Thank you.
Okay. So now, what are the gene switches? Why do you go from larva to pupa to adult? I mean, they seem to be different, completely different looking animals. And Williams and Kafatis, who was one of Williams' students and then became a professor at Harvard in very short time, and later went on to be head of the EMBO labs in Europe. proposed in 1971 that you have 3 different gene sets, the larva, the pupa, and the adult, and that you have a master regulatory gene for each stage. And they based it on bacterial gene control knowledge in 1971. Well, it turns out, what do we know today? It turns out that there are master genes, the master transcription factor. And we know at least 2 of them. Broad specifies the pupa, because if you have no broad, as I said, you get a larva. Don't turn on broad, you just molt to another larva. If you give JH to a pupa, you re-express broad. And I showed you that in Menduca, and this is what happens in Drosophila. If you re-express broad in the entire pupa, or I did it actually a little late, so the eyes, you do have some adult eye here, but the rest of it is mainly pupocuticle. And it turns out that there's another transcription factor in a completely different family that specifies the adult. And if you suppress that factor by RNAi, then you, in the pupa, this was first done in Drosophila, but it's also been done in beetles that are abolium and in cockroaches. Then you get a second pupa in the beetle and in the fly, and just another nymph in the cockroach. And both of these transcription factors, the E93 and broad, are important in chromatin remodeling of genes. And chromatin is basically the DNA with all the proteins around it, the histones and all the proteins that are wound up, because remember, you have a lot of DNA in your cells, but it's all wound up. And how it's wound up is you can wind it up such that you can completely enclose the gene so that nothing can get into it. So we call the closed chromatin structure. And what it turns out is that broad keeps chromatin in the pupal configuration, and you get the generation of the adult body plan. And E93 will close, it actually closes the chromatin around broad, so you can't be expressed anymore. And now it opens up the chromatin in other genes, so you get adult wings, for instance, and adult differentiation with wings. And so the question is, do you have such a thing, a master gene in the larva? And if so, what is it? It would have to turn on in the early embryo, in the egg. And people have thought that maybe it was croup homolog. But I told you that one of the things that juvenile hormone does is activate croup homolog. And croup homolog is there in the larva. But what the people did in Bombyx in the silkworm was they knocked down all the genes for JH production and action in the silk moth embryo. And they got no croup homolog when they knocked some of these genes down. And yet these went into the first instar larva, the second instar larva, and into the third. And then they didn't metamorphose, they just, well, they didn't do anything after that. So croup homolog has something to do with larval genes, but it can't be the specifier. So we go back to the fly again. And the only thing I want you to know here is that in the pupa, along with everything else I told you, the nervous system is remodeled. And my husband, Jim Truman, works on nervous systems and is very interested in how nervous systems remodeled during metamorphosis. That's one of his findings, and one that he's very interested in. And a paper came out in 2017 and that was talking about ecdysone induction of temporal gene expression in the Drosophila brain neuroblast to generate diversity. And what they showed was that there were 3 transcription factors. There was a factor called chemo, and then at the critical weight for metamorphosis in the last larval instar, broad came on in these neurons. And then after metamorphosis into the pupa, then E93 and caused late neuronal phenotypes. So the question is, and this we've been working on only for a year, because we got this idea, we were writing a review last year ago, and this really came out and started making sense. And Jim said, I had to show him a picture of you, and he said, well, we should have the one where we had our 50th wedding anniversary in July of 2020. And so this is our 50th wedding anniversary picture with a mask on. He thought that was useful. He does have a beard, but otherwise. Anyway, what we found is very fascinating, is that chemo is not just in the nervous system. Chemo is in leg imaginal disc. This is all in Drosophila. Chemo is in the leg imaginal disc. And then after critical weight for metamorphosis in the last larval instar, it starts disappearing. Broad comes up in the imaginal disc during the pupa stage, and then comes the E93. Now, in the larval tissue that's going to die in metamorphosis of the salivary gland, chemo is very high, and then in the last larval instar, it declines and disappears, and the glands die. Okay, well, so what happens if you remove chemo in these tissues? And it turns out you can do this in Drosophila, and I won't go into this complicated genetics, but it's just by crossing plies. And engrailed is a gene that's expressed in the posterior of every segment. And you can hook it up with a yeast transcription factor that will activate a red fluorescent protein, and you can also activate chemo RNAi in the same plot. And if you look at the imaginal disc here, what you see is that here is where the red is. So that tells you where the engrailed is being expressed and where the chemo RNAi is being expressed. And you see you have lost most of chemo staining, but you've in the imaginal disc, what you do is increase broad staining. But if you look at E93 in the disc, here is where the chemo RNAi is being expressed, but there's no E93. However, if you look at the larval tissues like the salivary glands, what you find is that broad isn't expressed, but E93 is in the where chemo is not. So you do the same type of experiment, but you look at the embryo, and here, let's see, here is where your RFP, where your engrailed is being expressed, so that chemo RNAi is gone, suppresses chemo there, and E93 comes up in this embryo. And what is really amazing is if you do this and you look at first instar larvae that are made by this, we get what we call tiger larvae. And so these stripes, every one of these stripes is where chemo is gone and E93 is coming up in larval epidermis. And what is happening is that normally when the animal, this animal is a large, extremely extra large first instar, it can't go to the second instar. And what you look at when you find underneath is that here's the L1 cuticle. Whoops. The L1 cuticle, L1 cuticle. Here is new L2 cuticle, but in the stripe region, the engrailed domain, the posterior part of the segment, there's no L2 cuticle. It's just a mesh, hash type of thing. And it doesn't make a new larval cuticle at all. And so if you look at the discs now, this is where the engrailed is being expressed. So no chemo is being expressed here in the control. And you see the red fluorescent protein, and chemo is around the outside, but not in the middle. And you look at, though, where you're exactly chemo RNAi, you can't see any chemo at all, and you get a mesh. And this red, and this disc is some of the cells are actually excluded from the disc, so it isn't a full disc. So what we're concluding then is that with the chemo is maybe the larval specification factor, that it comes on in the embryo and everything, the vaginal discs, everything are chemo. In a chemo null, the broad comes on in the imaginal disc in the larva, in the embryo, and the larval cells, like the larval epidermis or the salivary glands, E93 comes on. So you end up with a late embryo that can't hatch, or sometimes sort of comes out of the egg, but it has broad in its imaginal disc, which it shouldn't have until pupa, and it has E93, which it shouldn't have until the adult, in its larval cells. So what we think is that chemo is normally inhibiting broad and inhibiting E93 in the embryo and the early larva. And we don't know what genes it's turning on, but presumably larval genes. But it prevents the broad from turning on, and broad prevents, so there are no pupal genes turned on and no adult genes. So chemo is our larval specifier. And I thank you and my longtime collaborator and husband, Jim Truman, and all my graduate students and postdocs, including Kiyoshi. Kiyoshi did an awful lot of things for Manduka, and he never worked under Sofla, but he did a fantastic amount of the work on Manduka in the first finding all sorts of, and he worked on juvenile hormone in the black mutant larva as well. And then he did, when we'd started doing the molecular work, he was sort of the force in the lab. So thank you.
Thank you so much, Lynn. Oh my goodness, this is what's amazing is that this has been work that has been going on for over 100 years, and yet just in the last couple of years, you and your team have found all this amazing stuff about chemo and its role in controlling early larval stages. It's really quite remarkable how much is still being discovered even now.
Yeah, yeah. Well, as Jim, when he came to Harvard as a graduate student, said he thought everything in insect endocrinology had been found. That was in 1967. So, and it clearly wasn't.
Okay, well, now comes the time where everybody can unmute and it's free discussion. And I know that Jonathan Pelham is just chomping at the bit to ask some questions. So why don't you go ahead, John?
All right. Well, first off, I'm gonna, recommend Goethe. You cited him, and you are who you associate with, and I'm delighted you associate yourselves with us. That was incredible. In the language of the streets, you the bomb. All right. So just get that out of the way. I'm really fascinated on all of this. I'm a hanger-on, believe me, and it reminds me of all the conversations I had with Kiyoshi, where I was generally behind the curve. But I have a specific question, and I don't know if it has to relate in some way, but in checker spot butterflies, the larvae diapause in the middle instars. And before they enter diapause, they ecdyse. And then upon leaving diapause, they ecdyse again without feeding both times, right? So
Okay.
That's a very interesting thing. Does that have something to do with diapause?
Yeah. Now, I don't know about ones that diapause in the middle. I know that in larval diapause in, I'll think of it, the corn borer, no, not corn borer, but anyway, in Chippendale's moth, the larval diapause is enforced by keeping JH at an intermediate level. But they diapause and they go through what he calls immaculate molts, and then eventually the juvenile hormone level drops and they form a pupa.
Would you call that immaculate molt?
Immaculate molt because they don't have much pigment at that time. I haven't thought about this for a long time, but yeah, in the intermediate levels, I would assume that they molt, that it probably is a high JH, and high JH can be high enough to prevent the brain from releasing PTTH, particularly in the last instar. You can give JH, you could feed, like to Manduka, you can feed JH methoprene in the last larval instar, and they will go, instead of to 12 grams, they can go to 20 grams just by feeding them JH, and they won't molt because the JH is inhibiting the very high JH is inhibiting the PTTH from being released.
Yeah, I've seen some of those larvae. They're immense.
Yeah. So
one question that I have is you have a couple of models that you've used, the Drosophila and a couple of other specific insects. Is there work that's been done on other species, and do you expect those models to be applicable, or is it possible that there will be new models of metamorphosis?
I think that the basic for metamorphosis, I think the basic, it seems to work from Thermobia on up to Diptera. I mean, there are minor differences in how many imaginal discs you have, because beetles have fewer imaginal discs than Lepidoptera, and of course you have Drosophila and the higher flies with practically everything is coming from imaginal discs. So, but the the basic how hormones work on the metamorphosis seems to be the same all the way through. But in reproduction, that's where ecdysone, juvenile hormone, is important for ovarian maturation in many insects, but not all. And how it's important for ovarian maturation is different from insect to insect. I mean, I think it seemed to me that it depends on, you know, what the insect, how the insect interacts with its environment, and how the juvenile hormone is going to interact through that. You get into the social insects and it's slightly different, but juvenile hormone is also important for metamorphosis there.
What's interesting that some of the principles are basically across the board. I guess that reflects a successful evolution. And that brings me to another question about evolution of metamorphosis as an evolution, as a reproductive strategy, a successful reproductive strategy. Do you have any comments on that?
I think that reproductive strategies have evolved, you know, so that each type of it, or each, I don't want to say particular species, but each group of insects has their own, and they have to adapt to their particular environmental situation. And diapause is a good example, because diapause can happen in the embryo, in the larva, in the pupa, in the adult, and it depends on where they are as to what stage they might diapause in.
I guess it's interesting, you know, we think of growth and development, you know, in terms of mammals, and, you know, you're small and then you just get bigger and bigger and bigger. And yet on most of the planet, there's a completely different strategy of molting and metamorphosis. And it's just kind of crazy.
Yeah.
So another question is, I assume you're continuing your work on Kinmo. Can you give us a kind of insight into kind of where you're heading?
Well, right now we're trying to write the paper with the slides that I showed you. Basically, those are figures from the paper that we tomorrow are going to have some colleagues here at FHL, sort of a journal club that we have, except they're reading our paper and going to criticize it. And we're hoping to send it to Science. It probably won't get in, but at least, you know, this will be the first paper of trying to, we're actually doing work now on thermobia. We're gone all the way back to the fire brat. And it turns out chemo is very important in that system as well. And that is starting to give us some insights into the evolution of metamorphosis. And so I think that's where we'll be going, as well as Jim will continue to work on the nervous system, and then he's looking at nervous system evolution from the crustacea using mysids and several primitive crustacea up here in the summers when we can get them.
Well, Friday Harbor sounds like a good place for that.
Yeah. And we've been trying to do a little bit on, you know, we know that in crustacea that they have methylfarnesylate rather than juvenile hormone, which is just the precursor. Methylfarnesylate is the precursor of JH3. And that is what crustacea have, and they use it for reproduction. And in a couple of cases, they're supposed to use it for metamorphosis, but the metamorphosis is so different. So we've been trying to find out if it has any effect in mysids and it looks like it might, at first we thought it was going to have an effect in the embryo or the nopular stage. It looks like maybe it's later, it's between nopular and juvenile. But it's been difficult because we go catch the mysids and we work with them from about April to August, and then they disappear, and then we come back the next April. I mean, so, but we're getting so, I have a technician who's gotten very good at raising them in the lab, keeping them in the lab for 10 days or 2 weeks. And so that helps. And so I think we'll probably try to see if chemo goes down into the crustacea as well.
Yeah. So Bob, you have a question? Shoot, it's open discussion. So just chime right in.
I have a question and then a couple very short little stories. Lynn, is the same class of chemicals involved in the ability of crustacea and arachnida to regenerate, or at least crustacea to regenerate appendages, and the insect's ability or disability to do so? Inability to do so.
Yeah, that's complicated. In crustacea, the ecdysone is very important in regeneration, and the ecdysone is the same in the crustacea and the insect. I mean, it's the same thing. Of course, it's made by the Y organ rather than the prosthoracic glands, but it's used, and so you can get like in insect, I'm trying to think, Platella, the cockroach, you can get regeneration in the first half of the molting cycle of the nymph, of course, not in the adult, but in the nymph. And it all has to do with when ecdysone is being released. So what you do is delay the ecdysone release if you cut the leg off early in the molting cycle, if say day 1 or day 2 of feeding. And if you wait till day 6, then you don't delay and they don't regenerate. But that doesn't work in the Holobattalis insects. It's only, as far as I know, I'm trying to think. Oh, actually, and I don't remember all of this, but I think in Tribolium, you can get some regeneration in the larva. And I could send you a paper, because one of my former students is teaching at Wellesley, and he has just written a review on regeneration. So, and he's always been interested in that.
Well, thank you.
Yeah. Now, you had another question, I think.
Thank you. 2 little stories. First of all, when I was in high school, I don't know if my science teacher turned me onto it or if I saw a paper on my own, but I had read some of Carol Williams' Cecropia research. And in the summer of 63 or 64, I was raising a great mass batch of Cecropia. So when our science project came up that fall, I thought, well, I'm going to do some brains surgery on Cecropia caterpillars because I had a great abundance of them.
Yeah.
Well, I created, some really dead Cecropia. That's all. That's all on that one. It wasn't very successful, but it did tell me I was probably going to be an ecologist and not a physiologist. The second story asks a question of you. When did you begin your Manduka cultures at the UW in the lab?
Okay, we actually began them at Harvard. We brought them to in 73 was when we moved to Washington. We brought them with us.
Okay.
But I think actually Larry Gilbert at Northwestern had them in 1968.
Yeah.
And got them from there's a guy at the University of Illinois whose name escapes me right now, but he developed this tobacco hornworm diet and started raising them in the lab. You know, when I started out in Carol's lab, well, I started out in Carol Williams' lab as an undergraduate, and I injected tadpoles with juvenile hormone to see if I could inhibit metamorphosis. And you can imagine how many tadpoles I killed.
You had dead tadpoles too. Yeah, good.
Yeah, okay. Anyway, then I came back to Williams' lab in 1965 and spent a year when I was a protein chemist up until that point. And then I decided I'd change back to insects. And in that year, learned a lot of I tried all sorts of things with Cecropia. And one of the things was I decided, why don't we have a diet for Cecropia? Because the cherry leaves would, you know, there wouldn't be cherry leaves in Boston during the winter. So some guy at the University of Michigan had a diet that he claimed worked for Cecropia. And so I got it and I started feeding them, and they grew, but they grew very slowly. And then I started looking at the diet and discovered that the one thing they had it was a wheat germ diet. And so, but they didn't add any cholesterol. Now, wheat germ has some cholesterol, but of course insects can't make cholesterol, and they need cholesterol to make their molting hormone, ecdysone. So I added cholesterol, and I added beta cystosterol, which is a plant sterol. And then they grew, and I was raising Cecropia on diet all through the year. And it was an artificial, I mean, it was completely leaf-free. It wasn't like the Bombex diet that has mulberry leaves in it, but raising Cecropia on diet is not like raising Manduca on diet, because they got sick, you know, they get stressed. So I spent a lot of time, and raised Cecropia, Polyphemus, and all the Saturnids I could raise on the diet, but it wasn't very viable. And that's why I went to Manduca.
Yeah. Well, it reminds me of when I tried to rear some Polyphemus here through the winter. Nothing to eat in the winter here in Grays River, but I fed them finally on Salal, and amazingly I got most of them through, but they were the shrimpiest little Polyphemus in the spring.
Yeah.
Anyway, to go on from the Manduca question, so your labs, your cultures were way up and running in the early 80s. So here's the story, and it might relate to some winter night for you and Jim. In 1982, I was hired because the main insect person for Hollywood didn't want to come do the job. So I got hired to provide caterpillars for a motion picture written and directed by Michael Crichton and starring Tom Selleck. And it was being filmed in Eastern Washington. And the opening scene involved caterpillars and robots. Selleck was going to be a cop to keep robots in order. And it was a terrible spring, and Pelham and others tried to help me find caterpillars for my contract. We couldn't find any decent caterpillars in the early summer of 1982. And so I don't remember quite how, but I got onto your lab and I got a batch of Manduca. Were they sexta or Cuenca maculatus?
Sexta.
I thought they were sexta. And I don't know, they were surplus. They were no longer naive for the purposes they needed for or whatever. But I got about 100 of them. And you saved my life because I took them to the film site. Crichton didn't like it very much. He said they're green. And I said, well, they're caterpillars. And the director of photography said, oh, they'll shoot. Okay, they'll shoot. So they shot the movie. And so some winter's night, Jim get hold of this whole movie. It's called Runaway.
Runaway?
Runaway. And
Okay.
it's not bad. It's pretty bad, but it's fairly entertaining. Tom Selleck and several other substantial actors.
Okay.
Rather simple story, but in the second scene in the movie, there are mandukas from your lab
Okay.
in
We
a
will have
Hollywood
to see
film. I should have told you that years ago.
it. Great.
Okay, any other questions, comments, stories?
Nobody else. Let me ask you, did you get to actually work with Sir Vincent? I got to hear him speak in Cambridge once 50 years ago this month, and it was an amazing experience. Did you ever get to work with him?
I never got to work with him, but I did go talk to him several times. And when we went on sabbatical to Cambridge in 1986, 87, and we actually had lunch with him.
Oh, wonderful.
Yeah, so yes, he was really an amazing person, actually.
Yes, he was.
In what
As are
regard?
you.
Oh, thank you.
Thank you so much for speaking to us.
Oh, you're
Yes.
quite welcome.
Yes, thank you so much, Lynn. It's been extremely interesting. Really, really appreciate taking time out of your schedule to join us here tonight. So yeah, kudos, thank you.
Oh, you're quite welcome. I really enjoyed it.
Okay, thank you, everybody. We'll see you next month for our holiday party. So we'll keep you posted on that. Okay, good night, everyone. Thank you so much. Thank you, Lynn.
Good night.
Happy
Bye-bye.
holidays.